Hello, everybody. My name is Solana, and I will be going over the middle portion of this assignment and just giving a little bit more background on what strigolactones do.
My name is Matthew Hartman,
And I'm John.
and we are here today to talk to you about strigolactones, the latest in plant growth regulator discoveries and its effects on various plants. Since we are discussing a newly developed plant growth regulator, we felt it fitting to first include some supplementary information and perhaps some refreshers on what exactly are plant growth regulators. In humans, we have these funny little things called hormones. Hormones work to regulate all sorts of things in us, from growth to repair and reproduction. Well, plants work more or less the same, but instead of hormones, we have taken to calling these little chemicals and compounds plant growth regulators, or PGRs. Much like us, these chemicals work to regulate growth, development, repair, and reproduction, along with many other typical and specific plant processes. To follow the analogy in humans, we produce our hormones in specific areas called glands, and then have these compounds transported to other areas in the body. With plants, it is much in the same. Plant growth regulators are typically produced in areas where they are not necessarily utilized or expressed, and are then subsequently transported to areas where they are. This will be explained further in just a second. This transportation and production causes gradients to form during transportation, which in turn causes different physiological and biological reactions to occur based on the strength and concentration of that gradient based on its location. Plant growth regulators have significant value primarily in the world of tissue culture and plant propagation. Don't get us wrong, there is certainly no shadowing their importance in regards to the wild-type plants, but plants are pretty good at handling themselves without our help. So what are some examples of plant growth regulators? Again, I'm sure this is just a standard review for most of you, if not all of you, but this will be important later on, so please bear with us. First off, auxins. We should all be familiar with auxins at this point, but here's a quick rundown. Auxins are primarily produced in the shoot apical meristems and sent down towards the roots of the plants and the root area. Auxins are also regularly produced in the leaves, seeds, fruits, and other parts of the plant. Commercially, we find a lot of value in using auxins to induce rooting, especially in tissue culture, where plants usually don't have enough actual green material to produce these compounds on their own. Auxins also have vital roles in other plant propagation methods as well, as we all saw regarding kiwi root development. Auxins work to induce many processes in the plants, such as cell expansion, root initiation, vascular tissue differentiation, as well as bud and flower growth. Some examples of commonly used auxins include 2,4-dichlorophenoliacetic acid, 3-acetic acid, indole butyric acid, and naphthalenoacetic acid. Cytokinins are typically seen as the opposite to auxins in a vastly oversimplified and generalized sense. Cytokinins, as opposed to auxins, are produced in the root areas and roots of the plants and sent towards the shoot apical meristem. Their primary use is in tissue culture as a way to induce shoot development. They have far more vast uses in the plants themselves in roles that include cell division, chloroplast differentiation, as well as regulation of senescence. Senescence is a procedure plants undergo to break down its own cells to mobilize cellular nutrition to other parts of the plant. In other words, leaves turning orange and yellow in the fall is leaf senescence. Some examples of cytokinins often used include thidiazuron, which should be noted acts like a cytokinin but is technically not classified as a true cytokinin. Other examples of true cytokinins include zeatin, kinetin, and 6-benzylaminopurine, or BAP. There are many other plant growth regulators that we have failed to mention properly in the slide, but some others do include gibberellins, which are used for their stem elongation properties, asbestic acid, which is used for its moisture regulation properties, ethylene gas, which is typically responsible for maturation and ripening of fruits, and more. But these are all typically old news plant growth regulators, tried and tested over many years, and frankly, just not that interesting anymore. Today, we come to you with something quite special. We have to present before you one of the latest developments in plant growth regulator discoveries, strigolactones. Now, when I say new, it's obviously not completely new, considering its initial discovery was in 1966, especially since IBA, a very common oxen, and a very common rooting oxen, was only discovered in 1930, only 36 years prior. That being said, 1966 was only the initial discovery of Strigolactones, and was done through the roots of cotton plants. This was also where Strigolactones got their name, due to their well-known interaction with the parasitic weed Striga. Naturally, I don't feel the need to explain the very creative naming system that they decided to go with for this compound. The newness of this group of compounds comes from the fact that it wasn't even considered a true plant growth regulator until only in 2008. For all intents and purposes, we're going to use this as the latest date to base it as the newest plant growth regulator, mostly because we don't want to change our whole pitch for this presentation.
So, now that we know that strigolactones are plant growth regulators, what else do they do? Well, the name is derived from its discovery in the root exudates due to their ability to stimulate germination of seeds of the parasitic plant called Striga. Strigolactones are carotenoid-derived signaling molecules and plant growth hormones. Carotenoids are a pigment in plants, as well as an important source of metabolites with regulatory functions. Strigolactones have two main functions. First, as endogenous hormones to control plant development, and secondly, as components of root exudates to promote symbiotic interactions between plants and soil microbes. Some plants that are parasitic on other plants have established a third function, which is to stimulate germination of their seeds once in close proximity to the roots of a suitable host plant. Now, how are they able to do this? Strigolactones act upstream of these essential seed hormones to inhibit ABA synthesis and stimulate gibberellin accumulation. It is this ABA-gibberellin ratio that strigolactones regulate to break seed dormancy in both parasitic and non-parasitic plants. Signaling of these hormones allows for a symbiotic relationship between many species in the soil, including fungi, bacteria, and other microbes. The strigolactones are also a very important part in regulating several developmental growth processes in the shoots and roots of the plant. Now, though strigolactones are a type of PGR, there are also several types of strigolactones with several different structures. The strigolactones are a family of small organic compounds that contain a 4-ring structure. The ABC ring carries one lactone, and the D ring carries the other lactone. There are many different types of strigolactones, and different plant species may have different chemical structures and orientations. A few of the many types of strigolactone are strigolacetate, strigol, sorgolactone, and orobenchol. Strigol and orobenchol are all naturally occurring, but there's also other structures like GR24, which is a synthetically produced structure. They can also be classified into two subcategories, which are structurally distinct groups. There is canonical and non-canonical strigolactones. Canonical strigolactones contain the complete ABCD ring system, and non-canonical strigolactones are missing either the A, the B, or the C ring, but have the ional ether D ring, which is essential for biological activity. In some plant species, like sunflower and maize, non-canonical strigolactones in the root exudates are major germination stimulants. Though it has been found that there is a limited distribution of canonical strigolactones in the plant kingdom, this suggests that the plant hormones inhibiting shoot and branching are not canonical strigolactones, but they are rather also non-canonical strigolactones. What else do strigolactones do in terms of plant growth? Plants continuously extend their root and shoot systems through the action of meristems at their growing tips. By regulating which meristems are active, plants are able to adjust their body plans to suit local environment conditions. The strigolactones are produced in the roots and translocated to the shoots, where they inhibit shoot outgrowth and branching. Strigolactones work alongside other plant growth regulators to help inhibit this type of growth. The inhibition of lateral root and shoot growth allows for the plant to go taller as opposed to bushier. This is important because it allows the plant to grow taller and have better access to sunlight and other pollinators. The strigolactone mutants are unable to make or respond to strigolactones. This is also depicted on the image on this slide. The plant on the left is the wild-type plant that has functional strigolactone hormones and plant growth regulators, which is growing nice and tall with minimal lateral offshoots and roots. Now the plant on the right is a mutant that does not have or cannot activate the strigolactones, causing an abundance of many lateral offshoots and roots, causing the plant to be shorter. Levels of strigolactones, which are produced mainly by the roots, increase under low nitrogen and phosphate levels to regulate plant responses. The strigolactones that are produced in the roots get secreted more by the plant when it is under stress. More strigolactones will be released if it is nutrient deficient or stressed due to lack of water or any other external factors. The release of these strigolactones allows the plant to adapt to biotic or abiotic stresses in the soil. The signaling through the soil via the strigolactones also promotes symbiotic interactions between other plants, fungi, and soil microbes. Strigolactones also promote the elongation of seminal and primary roots, as well as adventitious roots, and they are suppressed in lateral root formation. This is advantageous for the plant because it is able to dig deeper into the soil and search for more water and nutrients, as opposed to growing horizontally or laterally.
Now, strigolactones play a significant role in the interactions between plants and fungi, particularly with mycorrhizal fungi. Mycorrhizal associations are mutually beneficial relationships between plants and fungi. Where the fungi assist the plants in nutrient uptake, the plants provide the fungi with carbohydrates. One of the primary functions of strigolactones is to act as a signaling molecule that attracts mycorrhizal fungi to plant roots. When a plant is in need of nutrients, particularly phosphorus, it releases strigolactones into the soil. These compounds serve as a chemical signal that attracts the mycorrhizal fungi towards the plant roots. Strigolactones facilitate the initiation of mycorrhizal associations by promoting the germination of fungal spores. In response to strigolactone signals, the fungi undergo a series of developmental changes, leading to the formation of hyphae that grow towards and into the plant root. You can see some examples of this in the images I've provided. Inside the plant root cells, the mycorrhizal fungi form structures called arbuscles. These structures are essential for nutrient exchange between the plant and the fungus. Strigolactones contribute to the development and maintenance of arbuscles, ensuring effective symbiotic relationships. The mycorrhizal association facilitated by strigolactones allows for efficient nutrient exchange between the plant and the fungus. The fungi enhance the plant's access to nutrients, particularly phosphorus, while the plant provides the fungi with carbohydrates produced through photosynthesis. Additionally, the fungi help prevent any other pathogenic fungi that wish to harm the plant from developing in the rhizosphere. Interestingly, the presence of mycorrhizal fungi can also influence the production of strigolactones in plants. This creates a feedback loop where the symbiotic relationship between the plant and the fungi is dynamically regulated. While strigolactones are primarily known for their role in the plant interactions with mycorrhizal fungi, there is emerging research suggesting that strigolactones can also play a role in interactions with bacteria. The interactions between strigolactones and bacteria are not as extensively studied as their interaction with fungi, but there are some insights. Strigolactones released by plant roots may act as signaling molecules in the rhizocube similarly as they do for the fungi. Some studies suggest that the strigolactones may influence the recruitment and activity of beneficial bacteria in the rhizosphere. These bacteria can contribute to nutrient cycling, plant growth promotion, and protection against pathogens. Strigolactones may also play a role in the modulating plant immune responses to bacterial infections. They can influence the balance between plant defense mechanisms and the establishment of beneficial associations with certain bacteria. Bacterial biofilm formation on a plant root is a complex process that can impact plant microbe interactions. While the direct influence of strigolactones on bacterial biofilm formation is not well elucidated, their role in root exudation and signaling could potentially affect the biofilm dynamics. It's important to note that the understanding of strigolactone interactions with bacteria is an evolving area of research, and more studies are needed to unravel the specific mechanisms and outcomes of these interactions. Strigolactones also play a significant role in the interaction between host plants and parasitic plants, specifically in the germination of parasitic plant seeds. These plants can include Striga, Orabonche, and Felipanche species. Depending on the strigolactones released by the host plants, they initiate these specific parasitic lifestyles. Parasitic plants produce large quantities of strigolactone-like compounds to mimic the host plant's signaling molecules. When these compounds are exuded into the soil, they serve as chemical signals for the germination of parasitic plant seeds. This process is known as strigolactone-induced germination. Once the parasitic plant seeds germinate in response to strigolactones, the emergent seedling develops a specialized structure called a hostorium. The hostorium acts as an attachment organ that penetrates the host plant's root, establishing a physical connection between the parasitic plant and the host. The parasitic plant's hostorium penetrates the host plant's vascular system and allowing it to extract water, nutrients, and carbohydrates directly from the host. This can lead to significant damage to the host plant, affecting its growth and productivity. Some parasitic plants are known to manipulate the strigolactone production of their hosts. They can induce host plants to produce more strigolactones, further promoting their own germination and establishment. The interaction between strigolactones and parasitic plants is often species-specific. Different parasitic plant species respond to specific strigolactones produced by their preferred hosts. This specificity is crucial for the success of parasitic plant-host interactions. Understanding this relationship can lead to the understanding of how parasitic plants can be stemmed and their infection halted. Now, why is this important? Why should you care about this other than being in a plant propagation class? If there is a plant that is a mutant and doesn't produce or respond to strigolactones, it will not form the beneficial fungi interactions, nor the bacteria interactions, and will be susceptible to pathogenic bacteria and fungi in the soil. Additionally, although the strigolactones does signal the germination of parasitic plants, without it, it will be increasingly more susceptible to parasitic infestation. Additionally, this can be used for commercial use. It can be used to enhance the root growth of cuttings. As you can see in the picture on the right, it's being used for olive tree branch cuttings and seeing how it can affect the root growth. Additionally, by forming these beneficial relationships with the mycorrhizal fungi, we could limit the amount of fertilizer we have to use and instead promote these beneficial interactions. Additionally, if we study more the relationship between strigolactones and parasitic germination, we could potentially find a way to stem germination of these parasitic plants or at least subdue them partially. Some of the major challenges associated with strigolactones is one, they share a pathway with all the other PGRs. So that means having direct control over it will be difficult, and influencing it will be tough without influencing other PGR pathways. Some of the specific effects that I discuss are species determinate, and we do not have a full understanding of how these effects can be generalized to all plant species, so further research needs to be done. And finally, it's expensive. Synthesizing SL, strigolactones, is an expensive process, and finding a cheaper alternative will be key for further research. Thank you very much for watching. Have a good day.